So today's speaker is Professor Wendy Scott Beane. Wendy did her PhD at Duke University in cellular, molecular, and developmental biology. Her doctoral work was entitled Building Gene Regulatory Networks in Development, Deploying Small GTPases. Currently, Wendy is a Presidential Innovation Professor and Associate Professor of Biological Sciences at Western Michigan University. Professor Beane has published a wide set of articles on topics ranging from the effect of magnetic fields on stem cell growth to the chemical composition of sea urchins. Today, we are thrilled to have Professor Beane with us to give a special seminar on quantum effects in biology. So with these words, Wendy, thank you very, very much for joining us. And we're really looking forward to this last seminar of this term.
Well, thank you. I am very excited to be here. I'm going to see if sharing my screen works correctly. Did that seem to work correctly on your end?
That's great.
Yay. Okay, we've done it. Anyway, thank you for that very nice introduction, and especially for inviting me to speak to you today. I love sharing my crazy little sphere of research with everyone. The title of my talk today is A Spin Dynamics Approach to Manipulating Tissue Growth. I was trained a little different, so I always begin with the ending bit. So basically, hopefully what you'll get by the end is that our work has demonstrated that weak magnetic fields in a field strength dependent manner can be used to modulate stem cell mediated growth in an organism. So in case you fall asleep between now and then, you've gotten the message and you're good. But to get everyone in a place where you can appreciate that statement or at least make your own determination about it, I like to go back to the beginning, which for my lab is the Planarian. It's a non-parasitic flatworm. We use the freshwater species, two of them which you can see here. They can be found almost anywhere in the silts of your favorite stream or lake bed, and they're super easy to care for. I don't know if you can see, I can't find my laser pointer, but the Tupperware here on the left, literally that's what we keep them in the lab. And adults, even of the same species, vary in size, but they typically range from a few millimeters to a few centimeters, so not very big. But they have several characteristics that make them really great for research, not the least of which is they have a complete central nervous system. It's predicted to be maybe the basal, the very first type of central nervous system that occurred, and they actually have a true brain, which, like your own, is bilobed. You can see it right here. More importantly, they have an exceedingly large population of pluripotent adult stem cells that are able to make pretty much every tissue in the body. The dividing stem cells you can see here, here's our planarian worm, our head to the left with the little eye spots, and all these green dots are the stem cells. Stem cells, in fact, comprise 25 to 30% of every single cell in the adult planarian. So all the research I'm going to show you today is done in adults. For comparison, only about 1% of your cells are adult stem cells. So it's obviously a big advantage for research. Now, planarians are hermaphroditic. They can reproduce sexually, but we're interested in them because they can also do this, which is reproduce asexually, which occurs through a process called transverse fissioning, which is just a really fancy way of saying the worm glues its butt down, it stretches itself really, really thin until it rips apart into two pieces, like you do. Process takes about 45 minutes. They like a little privacy. They like to do it in the dark. And both fragments are going to go on to regenerate into complete worms. So the head fragment is going to regenerate a new tail, and the tail fragment is going to regenerate a new head with a completely new brain. I've got a movie here showing a worm that has frequently done it. Here is the fission fragment, the little tail that's come off. And here you can see the head zooming by with its non-pointy end. But more importantly, this piece right here that is the tail, you'll note that it's lacking a head, but it's still moving. So this is no problem, at least for worms. So we use this ability in the lab to study how regeneration occurs. So say you decapitate a worm like they've done here, instead of dying like a sensible animal would, the worm instead goes, okay, and initiates the growth of new tissues, which you can see here in white. We call this the blastema, and this is the tissue that's going to go on to regenerate most of the missing tissue. Within 7 to 14 days, the worm is able to regenerate an entirely brand new head, including completely brand new brain again. And it doesn't really matter how many pieces you cut the worm into. Almost any piece of the planaria can regenerate into a whole worm. Each piece is going to form a complete worm with a complete central nervous system, even if the piece didn't have any piece of the central nervous system to begin with. So here is one worm on the left, which we have cut this fragment right to the right of it out of. So same fragment shown over 17 days. So this is all basically one worm through the process of regeneration. The brain primordia start to form within 24 hours. The eyes and brain are actually functional within 7 days. And the overall body shape occurs within 2 to 3 weeks. My lab is actually really interested in a lot of this process, especially how the new tissues know exactly what shape to form. But for the purposes of today, I'm going to concentrate on the first bit, what initiates the regenerative process in the first place. So that brings us to the real question. Why is a planarian stem cell researcher interested in quantum phenomena? Well, for a long time, I didn't realize I was, but the link comes from cell signaling downstream of free radicals and the ability of cells to sense their environment. So one such environment can be thought of as the Earth's natural geomagnetic field. It averages about 25 to 65 microtesla, depending on where you're standing. But weak electromagnetic fields are kind of present everywhere, including from the technology around us. Now, strong magnetic fields are really well known to affect chemical reaction rates in cells. We've known that for a while. And diagnostically, like here in this MRI, they can be very, very useful. But what about weak magnetic fields? So there's actually a lot of evidence that has accumulated that weak magnetic fields, so basically anything under a microtesla, can also affect cells. But it's historically been thought of in terms mostly of specialized cells that can sense quantum phenomena, sort of like the photosynthesis and the photon sensing you see here, or the migration of birds in the magnetoreception. But magnosensitivity actually may be a much more general feature of all cells. So here they show that when exposed to magnetic fields of less than 18 microtesla, so way less than the geomagnetic field, this decreases the growth rate of fibrosarcoma cells in culture. So basically a cancerous cell line doesn't grow as fast when you reduce the magnetic field. Another study used cultured rat skeletal muscles, and they showed that proliferation, or the cell division or growth rate, occurred much faster in magnetic fields that were slightly higher than Earth normal, which you see here on the control with the experimental on the right. So these effects are hypothesized to occur due to spin dynamics, which is where I come in. Magnetic fields can basically the radical pair mechanism, where magnetic fields drive either a singlet state, electrons having opposite directions, and this promotes recombination of free radicals, decreasing ROS concentrations. We'll get to ROS in a minute. But magnetic fields can also promote the triplet state, where electrons have similar spin directions, which promotes diffusion, right? And thus increases the concentration of free radicals. So what are we talking about? Well, this is an important mechanism or hypothesis because of one really important class of free radical reactive oxygen species, which I will call ROS. So the most biologically important for redox biology in cells are hydrogen peroxide, the superoxide anion, hydroxyl ion, largely hydrogen peroxide for signaling. Anyway, signaling in ROS occurs because an increase in the reactive oxygen species promotes changes in the cell, and it works in a threshold dependent manner. So stuff that's either too high, like too much ROS, or even too little, right, can lead to disease and even cell death. But that sweet Goldilocks zone right there in the middle, intermediate amounts, are known to regulate positively cell signaling. They can change the expression of genes, they control the migration of cells, they can control cell adhesion, and they can control, importantly for us, stem cell activities.
Sorry to interrupt you there,
Yes,
Wendy. So
please.
I've just had a question in the chat on the previous slide here.
Mhm.
It's a fellow who asked, could you please explain once again what signaling means in this context?
Okay, signaling in this context means reactive oxygen species interact with other molecules, usually in a thiol-dependent manner with a redox reaction, sort of transfer of electrons. And that causes changes in the downstream molecule that then goes on and does normal sort of cellular signaling, where one protein binds to another protein that either turns it on or turns it off, or modifies it to do such, which then leads to downstream changes in cell activities. Does that help? So
I think
usually in
that's
this,
clear, yeah, thanks.
okay, in non-quantum thinking, from biologist standpoint, ROS signaling is usually reduced to just talking about levels of hydrogen peroxide, even though those technically aren't a free radical, because hydrogen peroxide has many known downstream signaling partners that it can affect the activity of different proteins to control different cellular activities. Okay. Oh yeah, we were talking about stem cell activity. So this is where I'm really interested in. So there's a bunch of data showing that different levels of ROS have different effects on stem cells. So if I accidentally say the word proliferation, I really just mean cell division or making more cells. Very low levels of not none, these charts are kind of weird, but lower levels of ROS are known to keep stem cells in a stem cell-like state. So we call that self-renewal. Intermediate cells cause them to start turning into differentiated or mature cells that are going to have an actual function. So instead of a stem cell, it would actually end up being a sweat gland or an intestinal lining cell, that sort of thing. And they can even cause old stem cells to die if it's too high. So basically, controlling stem cell activities is known to be under the control of ROS levels. More recently, research has been shown that ROS are also actually required in a positive way for regeneration to occur in very many different species and the regeneration of many different organs. So a whole slew of them. Here we have a short representative. You have tadpole tails on the left, zebrafish fins in the middle, and axolotl or salamander tails at the end or to the right. And all of them are basically showing that if you lose ROS, it leads to a loss of new tissue growth or regeneration. So there's something about ROS signaling that is required for new growth to occur. Now, during Planarian regeneration, we found this same thing. I am on the left here going to be running a timeline of what's going on if you get lost. But basically, what we're seeing here is a general ROS labeling dye. So it's not labeling a specific species like superoxide or hydrogen peroxide. It's basically labeling anything that's interacting with an antioxidant, which is something that converts free radicals into something safer. you'll see several of these heat maps, so it's worth kind of orienting you. So anywhere it's blue, there's very low levels, and anywhere that it's sort of green, red, or white, there's very high levels. So what you see is after injury, within 30 to 45 minutes, you get this really large burst of ROS directly at the wound site. At the same time frame, the same half an hour to 1 hour, wound closure occurs. And this is basically where the skin cells come to meet to actually cover the opening to prevent the tissues from leaking out. A little bit later, 4 to 6 hours after injury, there is an organism-wide peak of stem cell division. So you can see this is sort of how much stem cell division was going on beforehand. And on the right, you can see there's much brighter red, which means that there's a significant increase in the number of stem cells that are reproducing or making more. And these are going to migrate to the wound site to start new tissue growth, which is sort of what we see here. So at 6 hours, that's also the time at which sort of the wound healing phase is complete and regeneration is considered to occur. This blastema formation, that new tissue, sort of peaks at the amount of tissue you get right around day 3 or 72 hours. So for the purposes of research, it's really nice that the very last thing that regenerates in planarians is their pigment cells. So all of this white tissue, if you see like this bright white tissue, that is brand new regenerated tissue. So it's very easy to tell if it's regenerating or not. So if you're following along, it's injury causes a burst of ROS at the wound site, which leads to a burst of stem cell proliferation, which is followed by new tissue growth. All right.
Okay,
so,
Wendy, I'm going
yes.
to interrupt you again just here.
Okay.
I know you're in the middle of something exciting, but we've had a question here from Dr. Alex Lasek in the chat. And he asks, has any of this research about stem cell control ever been applied to human stem cells?
Yes and no. So it's been applied to different types of stem cells in culture. I don't think it's been applied to stem cells like I would use worms in the lab. You know, I don't think we've actually had like human clinical trials or anything. I could be wrong. But definitely primary tissue culture, where you take tissues from patients and try them. We've done that. Well, not we, the field.
Okay, thank you, Wendy.
Sure. I'm not sure exactly what's going on is known. So it's kind of an exciting area. We know something's been going on for a while, and we're just really trying to parse down into exactly what's going on. The problem with human research, which is really interesting, is the fact that, remember, you only have 1% of your adult cells or stem cells. So it's actually kind of hard to find the stem cells and then to maintain them if you take them out of the body. Okay.
Interesting.
It is really interesting.
I'd never thought of that, but that is interesting. So
we think about it all the time.
If they're in the body, you can kind of keep track of them, but if not, you can't.
Sort of. There are some populations where we know there must be stem cells, but we actually haven't found what we call a niche, or niche. Still not sure if they're both okay or whatever. I hear them used equally. But basically, it's the environment that keeps them. Stem cells have to be special, right? They can't be like adult tissue. So embryonic stem cells are surrounded by other embryonic stem cells, none of which have really become mature tissue, right? So it's easier for them to maintain stemness, right? To be able to produce lots of different things. But if you think about it from an adult stem cell point of view, your adult stem cell is surrounded by a bunch of mature tissues that are putting out signals and chemicals and all sorts of things to try to make that adult tissue function in the adult. But you don't want the stem cell doing that, because you want it to be able to make more and to renew populations. So you have to, usually we think, create a special environment that protects it and helps it maintain its stemness.
Okay.
Yeah.
Interesting. Thanks, Wendy. And I've had another question here from Dr. Frederick Brazard. And he's writing, is the cell regeneration specific to sea animals, brackets, regrowth of brain? In this respect, does the pH influence regrowth, concentration of sea salt, etc.?
It works for land vertebrates as well as sea creatures. Both have been studied. Sea creatures, a lot of them are more regenerative, though please do not take that as canon. Things are regenerative at all different animal levels, and it's quite amazing what is and isn't regenerative. That's a whole nother seminar. pH is very hard. I studied pH a bit in regeneration during my postdoctoral studies, and organisms have such pH is so important. Organisms have huge buffering mechanisms to where it's actually very hard to change the internal pH by changing the external pH. Right? Because they have evolved to, you don't want to, if the external pH in, say, the sea or the ocean or your lakes or whatever drops or raises suddenly for whatever reason, right? If you respond to that instantly, you're less likely to survive, right? Than these mechanisms that have evolved over time to really buffer changes. So it's actually very hard to change pH during regeneration. So.
Are there any other like cool examples of animals that can regenerate their brain or?
Well, there's a bit of neuroregeneration going in all of us. So for a long time, it was thought that we couldn't regenerate brain cells, and we don't, but there are portions of our brain that can produce new neurons throughout your lifetime, though we're by no means certain, you know, we don't know all the story there. I don't know about brains specifically, but there are some very interesting mammals that have some really amazing regenerative abilities. So most mammals, like mice, are not terribly regenerative. They can regenerate their digit tips, which would be like losing the tip of your finger. But there is a species of, I believe it's African, the spiny mouse, Acomus, that actually has the ability to regenerate just huge, large portions of its skin. So if you've ever heard about lizards that will lose their tail, you know, as a defense mechanism, if a predator gets them, they'll just attach the tail and skittle off safe and then regrow the tail later. Well, these mice, for whatever reason, evolved the same thing, but with their skin. So if a bird or a predator grabs onto their skin is very loose, and the whole thing just completely rips off the back, like, you know, large, I don't know what the numbers are, but maybe like 50% of it or something on their back or more. And it's able to regrow the whole thing with no scar tissue. So, but most of the animal model systems are things like salamanders, newts, frogs, tadpoles, hydra, which are little polyps in the water. They're very tiny, zebrafish, that sort of thing.
Okay, thanks, Wendy. I have another question in the chat, but I'm going to keep that one for.
I told you this gets out of hand. I love answering questions, and it's all really cool.
I'm gonna keep this one for the discussion after your talk. So
I'm happy to talk to anyone ad nauseum about regeneration or Planarian in general. I love it. All right, so where were we? Oh yes, I was giving you background, and I was trying to hope to convince you that we had all the pieces to make my hypothesis make sense. So we know that weak magnetic fields should be able to affect spin dynamics, and we know that the spin dynamics will affect how much ROS there is. We know that ROS levels can change stem cell activity. And we also know from research that for some reason after injury, there's a burst of ROS accumulation that is required for new growth. So the evidence suggests that we should be able to connect the dots from the weak magnetic fields up here to new tissue growth and manipulate it through known processes or hypothesized processes. So therefore, we asked at the time what seemed a very seemingly simple question, like all simple questions was not simple at all. Can weight magnetic fields be used as a tool to control stem activity during regeneration in an animal, not in cell culture, but actually in the organism when it's normally undergoing it? So our collaborators got to work first teaching us basic physics because all of us had forgotten it. And along with that, they created our mu metal mag shield box, which you can see here on the left. Mu metal blocks all magnetic fields. So that gets us near zero. And by placing Helmholtz coils inside, we can experimentally produce a fairly steady static weak magnetic field of pretty much any given strength for anything under a microtesla. We typically run Earth normal controls on one side of the mu metal partition, and then our experimental field strengths on the right side at the same time. So we thought, hey, why don't we just amputate some planarians? So we removed head and a tail like we've done here. So the red lines are the amputation plans, creating trunk fragments. You can see a fragment there 3 days later. And we just let them regenerate in the mag shield box for 3 days at different exposures. We found which was pretty cool, that exposure to static weak magnetic fields was indeed able to regulate regeneration. You probably saw that one coming. We exposed regeneratives to a whole range of field strengths from near zero up to 900 microtesla. And we looked at the level of regeneration or new tissue growth at 1 hour after injury. So as predicted by the radical pair mechanism, where the spin dynamics can change the ROS in either direction, we found that different weak magnetic field strengths could cause opposite effects in tissue growth, with some reducing the amount of regenerative tissue and some increasing it here in green, which may not seem like a lot to you, but biologists really like linear relationships, and this caused us some ulcers for a little while until the physicists convinced us that this was a good thing. So for the rest of this talk, we're basically going to concentrate on just 2 field strengths. One that inhibited the growth. You can see here at 200 microtesla, we've got our control level of new tissue growth. Remember the white blastema. And then on the right, we have sort of the activation of the system at 500 microtesla, where we can get significantly increased amount of new tissue that regenerates. So we thought this was occurring to ROS. So obviously we went then and looked at ROS accumulation at the same near 0 to 900 microtesla range. And again, we found that when exposed to different weak magnetic field strengths, this resulted in opposite effects on ROS levels. And I'm happy to talk to you about internal feedback mechanisms and why the 2 charts don't look exactly alike, but we'll never get to the end if I do that now. So here,
when I've just had a very quick thing. So someone in the chat asked, can you remind us what the Earth's magnetic field is again?
Oh, yes, I am sorry.
That is the 45 microtesla, right?
Yes, so I should have explained that. So in our quote controls here, we're exposing them all to basically Earth normal. We also did untreated ones that weren't exposed at all, and they all came out exactly like the 45s with no statistical differences pretty much at all ever. As far as we can tell. So these graphs are basically change from Earth normal, if we take Earth normal to be an average of 45. So you'll see 45 sort of control in the middle, with 200 being a reduction and 500 being an increase. Does that help? I'm sorry, I should have explained that earlier.
No, but I think that's clear. Thanks.
Yes,
perfect.
Great. Okay, so basically we saw the same thing with ROS, but in much prettier pictures. So in the middle is that burst of ROS that occurs just at the wound site. If you expose it to 200 microtesla, we do a really good job at eliminating it. And conversely, if we expose to 500 microtesla, we significantly increase the amount of ROS pretty much everywhere. All right, so what's so important about ROS? So we wanted to find out a little bit about what ROS was actually doing from a biologist standpoint. Applying magnetic fields and seeing effects is a bit phenomenological, so we kind of want to know a mechanism of what's going on. And we found that ROS accumulation is required for this expression of this gene called Hsp70. Heat shock protein 70. It's just a stress response, chaperone protein. It has a lot of different functions. It doesn't actually kind of really matter what it does, but we know that changes in ROS can affect the expression of this gene, whether it's being produced in a cell or not. So what we have here is RNAi, which is just a mechanism to knock out the expression of a gene, so the tissues don't have it. The probe up here in blue would be where HSP is, and the ones on the, so those are our controls, and the ones on the bottom are where we are also looking for HSP expression, but here we're checking our control. So if we knock out Hsp70 with the RNAi, we lose expression of Hsp70, which is a good thing, because if we don't, we're in trouble. But more importantly is this bottom right panel, where if we use a drug, DPI, doesn't matter what the drug is, but it causes a loss of ROS. You lose expression of Hsp70. So down here is our growing pathway. Injury to ROS accumulation to the expression of this gene Hsp70. Now, Hsp70 is important because it is what's required for stem cell division, for that increase of stem cell growth that you see after injury. So here, if you lose Hsp70, this is what the normal upregulation of stem cell is after injury. And this is what happens when you block Hsp70. You lose the stem cell response. So injury, ROS to Hsp70 to stem cell proliferation. Oh, sorry, division. Okay, so that stem cell response is what results in the production of the new tissue. So if you lose Hsp70 because you've lost the stem cell response, you also subsequently lose the RNAi. You've also lose the new tissue growth. So basically, long story short, what we show is that ROS is required to turn on this gene Hsp70, which is required for the stem cell response to injury that leads to the new tissue to grow. So here is our little pathway of what ROS is doing in planarians following, like when we cut its head off, like we've done here. Okay, but what about the weak magnetic fields? So this is a recap of the data I showed you a couple slides ago, because I don't know about you, but I never remember a couple slides ago. So here they are again. I showed you that we can play around with weak magnetic field strengths, and then we can either increase or decrease ROS levels, and we can increase or decrease the amount of new tissue that's grown. But is this occurring through that mechanism that we saw before? And so basically in an Hsp70 dependent manner, right? So is it going through this same pathway? And you're probably also not surprised to hear that the answer was yes. We found that at 200, so here's our control Earth normal in the middle. So this is what it should look like after injury, where we have more Hsp70 and more stem cell division going on. 200 microtesla reduces both the expression of the gene and the stem cell division that it controls, while 500 does the opposite. It increases the expression of the gene and the amount of stem cell division. So basically, we can take our pathway, and it looks as if the data suggest that we should be able to affect this whole pathway just by manipulating weak magnetic field exposure. So it suggests that the effects on stem cell activity can be explained largely by altered ROS levels. Okay, so we have a pathway We know that weak magnetic fields can affect growth by changing ROS. We know that it varies by field strength, whether it goes up or down in the amount of regeneration. But again, so we found a way to control Planarian stem cells. Is there really a point other than possibly starting a little Planarian A and E, which I love that idea and wish I could. But if we want to apply these findings to other animals, maybe even humans one day, one of the first things we needed to do was to determine whether these effects are predicted. Are these just kind of random effects, or do they respond in a way that can be used therapeutically, right? So our data suggests that we should get the same results if we directly manipulate ROS that we got when we manipulated the weak magnetic fields. So in fact, that's what the data shows. We see that if we directly block ROS with that pharmacological inhibitor, you not only lose ROS, which we should, right? That's a control. We can reproduce the same loss of Hsp70, which I'm not showing you here, but we have that same reduction in stem cell division and the same loss of new tissue growth. So basically, inhibiting ROS directly replicates exposing them to 200 microtesla. So we would predict the opposite if we increased ROS directly, like we saw with the 500 microtesla exposure. So we used a couple different mechanisms. We knocked out a gene, SOD, superoxide dismutase. It basically just breaks down superoxide into hydrogen peroxide. So if you delete that gene, you get a lot more superoxide. So we get a big burst increase of ROS. And this leads also to an increase in Hsp70 and stem cell division, and an increase in new tissue growth. And we see the same effects. I didn't have room for all the data. There's just too much data. But basically, we see the same effects with the with knocking out this enzyme that produces disrupts the ability of the cell to break down ROS, as if we treat the cells exogenously from the outside with ROS, in this case, hydrogen peroxide, where we can get an increase in proliferation and new tissue growth. Okay, I don't know if I'm going to get through this. We might have to skip this. Okay. So the point here is basically, if we want to translate this into, this is good news that it's predictable, because we would want to be able to manipulate it in a non-stochastic way if we want to translate it into therapies. And there's a lot of potential for being able to non-invasively regulate ROS concentration at wound sites. So this slide is basically data showing you that we've known for a long time that ROS signaling is a very important part of the wound healing process, especially in humans, but even in animals that, even animals that regenerate really well, like zebrafish, you get a spike of ROS at the wound site attached to just healing of the wound, right? And then you get a later spike of ROS that's associated with regeneration. This later spike does not occur in humans. Okay, so in planarians, my graduate students, on sort of a side note, in my more biological aspect, found that ROS is required for wound closure. So basically, you have a bunch of wounds. You can see tissues coming out in controls, and then by an hour, well, really, before an hour, it's closed. There's no more tissue coming out. Nothing's leaking. So that's wound closure. If you block ROS, that never closes, right? It blocks the abilities to wound to close. So tissue keeps coming out. And we found that that worked by controlling, well, if you care, actin-mediated changes at the wound edge, but basically the ability of the skin cells to cover the opened wound. So ROS is directly required for wound closure. And here's where it gets interesting. So we found that different levels of ROS occur in wounds that just heal, meaning they close up, right? As opposed to wounds that regrow new tissue or regenerating. So even if you account for different overall wound sizes, because healing wounds are usually smaller than regenerative wounds, but even when you factor in a ratio, wounds that do not form new tissue have lesser amounts of ROS at the wound site than regenerating. They still have more than the amount that they had before they were injured, but not as much as if they're going to regenerate. And on top of that, we can use levels, different levels of ROS to control whether we want a wound to heal or to regenerate. This is a really exciting part for me. And I want to repeat this with the weak magnetic field stuff. It's in our future stuff. But basically, if you go down here to the second row, you will see an ROS inhibited. It says DPI, but it's a loss of ROS. And you'll see that the wounds don't close, there's tissue coming out, and it doesn't form a blastema or anything. It just sort of kind of seals up. And you can see that sort of here. Okay. At 200 micromolar, oh, I'm sorry, was there a question? Oh, okay.
No, I think you're fine. Sorry.
Okay. Yeah. Okay, no, that's fine.
Cheers.
So then if ROS, controlling the level of ROS at the wound site can control the outcome, then we should be able to give it different levels of ROS after we've inhibited it. So we inhibit the natural ROS that occurs after injury, and then we add back hydrogen peroxide from the outside, okay? If we add in 200 micromolar, it doesn't do anything. It does exactly like it did when it was inhibited, because it's not enough to compensate for the drug that's inhibiting the ROS. So it still leaks and it doesn't heal, it doesn't regenerate. If we add slightly more, 300 micromolar of hydrogen peroxide, it heals great. So that's what this first blue spike on the top graph is, but it does not regenerate. So if you look at the bottom graph, it's still a red small bar because it's not regenerating. So at this level of 300 micromolar, we can promote wound closure, but not new tissue growth. But if you add more, 400 micromolar, we both close wounds, like we see up here, as well as grow new tissue. So this is where it gets super, super exciting, because now we might be able to use weak magnetic fields to manipulate stem cell activity by carefully manipulating different levels of ROS at the wound site. And this would be really good for cancer and regenerative therapies. Okay, so future directions. Very quickly, some data we just got this week, so it probably doesn't look as great. But what I've been showing you before is this top dye, general ROS dye. It does thiol interactions, and basically anything that's an ROS, it will fluoresce if it's there. But we wanted to start thinking about what was going on, what exactly in these reactive oxygen species is the magnetic field affecting that is responding or sensing to its magnetic environment. And so the first thing we looked at, obviously, at least for biologists, was hydrogen peroxide. And we do see a spike of hydrogen peroxide at 1 hour when we see the spike of the general dye. That dies down a bit, still up, but dies down. So that's good. So this is a hydrogen peroxide specific dye. But then it gets weird. So when we apply weak magnetic fields, which we know affect the general reactive oxygen species dye, so all reactive oxygen species together, it can affect, and we know it can affect new tissue growth and stem cell activity. When we look at the hydrogen peroxide specific dye, we see absolutely no change at that 1 hour mark with our magnetic field. So 200 microtesla are Earth normal 45 controls, and the 500 microtesla all have statistically insignificantly different levels of hydrogen peroxide at the wound, which opens up crazy cool thing. So this is where everything's going. So I was trying to find a better picture of this. We also have data to support it, but basically we think that it may be affecting the weak magnetic fields, may be affecting superoxide, because we have slightly different phenotypes when we block ROS with this superoxide dismutase, the SOD that I told you about, rather than messing around with hydrogen peroxide itself. One of which is that the superoxide is upstream of that Hsp70 expression, where changes in hydrogen peroxide are not. It's the one thing that we found is very different. So we have a lot of stuff, fun stuff to do in the future. Also, when making this, I realized that thanks to the pandemic, I don't actually have a picture of my lab. This was the last one I could find from 2019, literally the Christmas before it all happened. We should really rectify that. Here are our current members. Here's alumni, our engineering collaborators at the University of Colorado, Frank Barnes. Two main graduate students did a lot of this, though pretty much everyone in the lab helped, but Alana Van Heusen right here on the left, and this gentleman in the middle of the field of women for a while. I only had one male in my lab, it's very weird. Luke Kinsey, those two did the bulk of the work that I have showed you today. And thank you to my funding sources, largely the NSF. Who have been very kind to me. Thank you.
Thank you very much, Wendy, for an extremely clear and super intriguing presentation. If anyone has any questions, I can see they've already dropped into the chat here. Just please asking questions. Please keep asking questions. I'll try my best to moderate them. I see that Crispin, you already have your hand up, so go ahead.
Okay, so firstly, my deep apologies for missing the first few minutes of your talk.
Fine.
I was at the head of the department's office. Anyway, wonderful talk. Thanks very much. Very interesting. One of the things that I've been reading recently is about controlling insulin, the effect of insulin on sugar in with magnetic and electric fields. And I wondered whether you thought about that as a possible mechanism here, and whether you looked at electric fields specifically to see whether they have the same sort of effect.
Yes and no is the answer. I have thought about pretty much anything I've come across, since I have no idea this is like a whole new world for me in the last 4 or 5 years. But I have thought about a lot of things. I do have some problems with I don't know if you want me to stop sharing my screen or whatever. We don't have a vasculature, right? So we're not going to regulate, not we have a vasculature, right? But the worms don't. All of their cells are connected by an intestinal tract that takes care of all of basically the functions that your circulatory system does, except for gas exchange. The reason flatworms are flat is because they directly diffuse oxygen from the outside down, and they could only be so thick because that's how far the oxygen can get through the cell layer until it won't reach. They don't exactly have the same mechanism for dispersal, though they do have immune responses, largely innate, and they do have hormones and things, except we found insulin receptors, but I wasn't able to find an insulin homolog, but that doesn't mean it doesn't have one. It just means I couldn't find it. We have a really well annotated genome, so that's not the problem. But sometimes basal forms of genes are slightly different enough that they're really hard to recognize at first, and sometimes they're not conserved in sequence as much as function. So sometimes you're looking for a gene that's a different sequence but has the same function, which makes it a lot harder to find. Yes, I think it probably plays a role. Something else I'm very interested in is that there are very interesting data out there about the effects of reactive oxygen species on neutrophils and certain diseases that are immune related, and the abilities of weak magnetic fields to affect the ROS in those cells. I'm not sure we know, like all of this, these are all kind of new fields. So we know a bit to get really excited, but not enough to really explain a lot of things, which for me is the fun part, right? Like I want to go in and figure out what's going on.
Brilliant. Okay.
I don't know if that answered the question, but
it's just that I was thinking about wound healing and sugar, the control of sugar.
I don't know that they have the same response that we do. I also don't know that they don't. I don't know if anyone's bothered to look at sugar metabolism after eating in planarians. It's not exactly a species you'd think to do that in, you know what I mean? Like, honestly, I was interested in stem cells and how tissues know what to become, right? So I chose the Planarian because it's a great model system for that, because they're plastic. They have a lot of ability to change, right? And then I got sidetracked into what's now mostly in my lab, which is ROS signaling and this quantum phenomenon. But if I was going to do it the right way and think, oh, what model system should I be looking at to be studying quantum phenomenon? I think I'd put Planaria down at the bottom. There's a lot of technical challenges in dealing with an aquatic animal that's covered in mucus, that doesn't have a circulatory system. You know,
it'd be a bit much to feed them and then chop their heads off. Anyway,
yeah.
Anyway, I should let other people ask questions.
Sorry. Yeah.
Okay. I think we have we had several questions from Dr. Brozard. So I promoted you to panelist. Frederick, are you with us on the call?
Yes, can you hear me?
I can.
Oh, okay, great. Thanks, Wendy, for your presentation. Very good. I mean, it's been most of it has been clear to me, which I didn't expect that, being non-expert. I was wondering, so you show one of the graph where you show this effect on the ROS at 200 microtesla. Then you have a negative effect for this, and then you have a positive effect 500, and then again it seems to go down for 900. Do you know why there's this kind of trend?
Yes.
For 900.
So wait, hold on. That's the Blastema size. So, well, I say yes, but it has to do with a level of math that I, as a developmental biologist, was hoping to avoid in my career. But basically, because of the mechanics of the spin dynamics, it's basically on or off. So it would be actually predicted that we wouldn't see like slightly more gives you slightly more, right? So, which is what I expected to see. I figured if there was a field effect, a little bit would give you one, and more would give you a different effect, and then more would give you more of that effect, and whatever, right? That there would be a linear relationship. And with the ROS, working on a threshold, it's more of a, you either have enough to start that signaling pathway or you do not, right? So changing the rates of recombination, again, this is all consistent with, we don't actually have like, what we're hoping is to be able to measure spins in the organism itself, but it's consistent with it, right? So we would either be promoting diffusion, right? So we're making more radicals. So we should be increasing everything, or we should be promoting recombination, so that we lose our free radicals and our signaling goes down and we lose things. So it makes sense with the mechanism. What's interesting for me is this spike right here. So if we go back up to 900 microtesla, we see a spike in ROS that is statistically significant. But if you'll watch your eyes for a second, here's the chart for growth, right? So one of the reasons we looked at 200 and 500 is because they were in the same direction on both graphs, and most directions are. But you'll see at 900, there's no statistical difference. And what we're sort of seeing, which makes perfect sense biologically, is that there is a delay. Because there are buffering and feedback mechanisms to make sure you don't have too much ROS, which is going to lead to cell death, right? Too much ROS will program cell death and kill off cells. That's one of the ways cells regulate themselves if they have like damaged DNA or something. So it would make sense that you would change statistically the levels of ROS in the cell before you would statistically see an effect at the end of regeneration, which is what this blastema or new growth chart is.
And
yeah,
thanks. And have you studied this with enzymes? The effect, because we have done some work. I mean, not me personally, but through collaboration on artificial enzymes, and we found that the hydrogen peroxide plays a crucial role there as well in the activity of the enzyme. And
yeah, so I have a really long lifetime list. I need at least 3 of me, and I need to be 20 again to get everything done that I want. But part of my goal in going into this at the beginning was I felt like a lot of these things have been being studied by engineers and physicists for a really long time, right? And things like traditional signaling with reactive oxygen species have been studied by developmental biologists, but the two fields aren't really talking to each other, right? And there's a lot of data in both fields that each of us doesn't know about, right? And we're not connecting the dots. And so what I really wanted in my, really not great model system, but the one I work with, right, is to sort of take the theory, and someone who hopefully knows the theory, and then try to connect all the molecular dots for one process, so that we could basically say in a whole organism from start to finish, we see that the magnetic field causes this effect, which is responsible for this effect, which is responsible for this effect. And I think the goal for that, if we concentrate on the details of one specific pathway, is the hope is to get more buy-in from more biologists, because there's a lot of data, especially from the Russians, I think in the 60s or 70s, showing effects on weak magnetic fields on tons of animals, and specifically lots in Planarian, and some really interesting data. But it's basically like we exposed it to some magnetic fields, and we saw these, they grew, or they didn't grow, or they got longer, or they got bigger, or whatever, right? But there's no molecular stuff in connecting the two, and there's a real bias in the developmental biology world that if you don't know the molecular mechanisms that are going on, then it's not really, I don't want to say not real, but you know what I mean? They're just like, you saw an effect, who knows if it's really, biologically important for the animal.
Yeah, okay, thank you very much.
Yeah, I definitely want to get into enzyme work though. Sorry.
Okay. Thanks a lot, Frederick and Wendy. We have 2 questions from Alex. One is marked as a fun question. I'll start.
Oh, good.
With the fun question is: can you manipulate the flatworm to have 2 brains in one body at the same time?
Oh yeah, I should have put it in here. I've done it up to 4, but I did it with a pharmacological drug, not weak magnetic fields. So
wow.
But I might be able to. We could do that. But yeah, that's
it's like a mini Frankenstein's monster somewhere in the lab.
It really is. And the problem with multi-headed planarians is the heads don't talk to each other. And the way planarians eat is they have a lot of chemosensory neurons along their lateral edges, and they basically sense the water to figure out where the food is, and they go towards it. But the problem is when you have more than one head, they each want to go in a different direction. And they don't get anywhere. And so they tend to, over time, starve to death.
Right. So yeah, so the Greeks had it right when the two-headed creatures didn't last very long.
Yeah, and two heads are not better than one, apparently, if you're a planarian.
Okay, so that's Alex's fun question. Then I suppose this next one is possibly the boring one then, which is, but I don't think so. It's maybe the more kind of scientifically interesting one. So you mentioned applications to cancer. So how is this related to cancer regeneration or healing, etc.?
Okay, so one of the things that we need to do, I just need more personnel, is to really start investigating. One of the reasons why I haven't done all these cool other things is because one of the things we really need to do is to establish if we are affecting all cells, and it just happens to be that stem cells are more sensitive to ROS, or if we're only affecting stem cells, or what, right? Because it looks to us at a preliminary investigation, but we certainly haven't done a thorough investigative search, that we're really not affecting pretty much anything except the regenerative program, right? And that the rest of the planaria isn't bigger or misshapen or dying or growing out of control or whatever, all right? So basically, we have a cell that is undergoing division, proliferation, or growth, whatever you want to call it. And we have a way with weak magnetic fields to non-invasively control that growth, right? If this is true, if it is a specific to cells that are undergoing division, right? These ROS signals, then it could be. So what's the problem with cancer treatments now, right? They generally just kill everything, right? And you try to treat just the tissue, but you kill lots of healthy tissue along with it. So most of the horrible, chemotherapy and radiation effects are from killing off the things you didn't want to kill, right? What if we could, without going in and doing surgery, apply magnetic field of some strength or some sort and just affect the growth rates of the stuff that we want without affecting the other tissue? This is like decades in the future. We're not saying that now, but that's how it relates to cancer. Because if we are dealing with cell growth and we can make cells grow or not grow by changing ROS levels, then it's not just a regenerative medicine thing where we could maybe apply it to a wound, right, and encourage it to regrow. Like if you cut off your thumb or something, could you regrow your thumb? But also maybe we could apply it to tumors and reduce the size of tumors. That's kind of the ice cream goal at the end, maybe someday. Interestingly, I think our data is really suggesting that we really need to investigate this more, if only because, like if you go to A&E and you have a wound, they're going to clean it, most likely with hydrogen peroxide, most likely at a concentration that we know will inhibit regenerative growth in pretty much all the animals that we've done, right? And so it's not that they shouldn't be doing that, because it does a really good job of, making sure your wound closes, right? And doesn't get infected. But if we also want to encourage regenerative medicine, we have to have a better understanding about when and where and how much of these signals are required.
Cool. I think Frederick, then Hugo.
Again, thanks. I was wondering, did you find a correlation between the size of the worm and the magnetic field strength you had to apply for regrowth?
No, largely because unlike pretty much every animal, there isn't a standard range for planarians. Planarians basically, and they, all right, so most things, like if you have an embryo or a small child, it will be in a given range for its age, right? Like whatever species you have, right? If you've got a horse, you don't expect the newborn foal to be as big as the horse, right? And as the horse ages, you don't expect it to get small again, like the horse, like the foal, right? So planarians have, one of the reasons I study them is because of their cellular or tissue plasticity. They can not only undergo growth where they eat and get bigger, but they can do this thing that we've called unimaginatively degrowth. So pretty much if you starve anybody, human, dog, planaria, you end up using your own cells for energy, right? In pretty much every other animal that we know of, right, that leads to you looking like you're starving to death, right? You can starve a human for a month or so or whatever, however long, and you can tell it's been starved, but it isn't necessarily shorter. It doesn't look like a child. It looks like an adult that has been starved. Now, if you starve a planarian, it can go for your typical one centimeter worm or so can go like 4 months without food, using its own body for energy, right, and nutrients, except it will degrow proportionally. So at any point in time, oh, I don't have any of my degrowth ones. I usually put a degrowth in here because it's cool, but I was trying not to go over time, which I've done anyway. Sorry. No, I can't find, oh, these are all my other, these are my backloaded slides. I don't have it in there either. Sorry. So I showed you the chart of, you know, sort of if you had a smaller worm and a larger worm and an even larger worm, right? The middle worm, that's the middle size, there's no way to know if that is a young worm that is eating and getting bigger or a full-grown adult worm that's been starving and getting smaller. There's no way to look at it and tell because it proportionally rearranges all of its tissues to look normal, just smaller. So we have to do all of our calculations based on proportional by sizes, right? Because if we're looking at amount of ROS in a bigger worm, it would have more ROS. So we have to control for worm size. So.
Thank you. But I guess if you were to study, for example, tentacles of an octopus, which also regrow and which have their own brain, that would be interesting. But you would probably need a stronger magnetic field. You would.
I don't know. The way I got into this is so remarkably unlikely. It's amusing, right? So I ran into Frank Barnes. They invited me to give a talk at BioEM, which is a Bioelectromagnetic Society meeting. And I thought they had the wrong person, but they said, no, they didn't. They wanted me to give a talk. So I came because it was a free trip to South Africa. I'm going to go to Cape Town if you invite me. He said, this is exciting. I want you to come to Colorado. You can give a talk and we'll try some of your worms in his, well, basically the mu metal, that mag shield box I showed you, right? He had some that he had built for his own, where he was doing, working with someone who did cell culture. So that cell culture data I showed you, some of that was his. And he had been having effects at 200 microtesla in cells, in human cell cultures. And so I was there for 3 days. I shipped the, I put the worms in my carry-on on the plane to see if I could sneak them on, and I did. This is like pre-COVID, got them there, cut them up under really sort of not very careful conditions, stuck them in the box, and then I had to leave before they were ready. And so they shipped them back in the mail. And so what should have happened with that whole thing is that they all should have died and we should have gotten nothing. And instead we got a bunch of worms that didn't regenerate and a bunch of controls that did. So I'm thinking that this may be a function of eukaryotic cells, not necessarily the organism that they're in. But that's only my, personal hypothesis.
Okay, I will shut up now. Thank you.
No, don't. The problem is getting me to shut up. Okay, so you're gonna have to put a pin in it when everyone wants to run away.
So I think Hugo's next, and then I have a quick question from Chris, and then we have Pierre with a question that's gonna have to be the final question of the seminar.
So yeah, this is a really fun talk, and I guess there are lots of questions that can be had. The question I had, so all these magnetic fields that you apply, these are static magnetic fields that just have the same. Have you looked at oscillating magnetic fields to see like this will just
so
limit
frequency adds a whole nother wonderful people have, I have not. That's the short answer. Okay, so in cell culture and other people have shoved planarians under oscillating fields, but haven't been looking at it molecularly like I have. Most of that's still, you know, decades ago. Part of our problem was looking at it back now, it took us a stupidly long amount of time to understand how to work a very simple Helmholtz coil. Like, and we were on the phone with the engineers in Colorado going, I don't understand, you know what I mean? Like, it's just, you know, it's like if I shoved you into a biological experiment and just said, go, you know what I mean? Even easy stuff would be a little, you know, confusing at first, right? So I really, really, really want to get this set up for the AC, but I don't want to set it up myself. And so this summer, I'm trying to get my graduate student before he gets his job he's applied for, his tenure track job, to go out there and have the engineers, you know, walk them through it or build it there and ship it back or something. So, but yeah, we're going to get much better effects with frequency.
Okay. Do you have a hypothesis on what you expect much better to mean?
I think, well, the initial hypothesis was that you would get resonance with, say, calcium. There's a lot of cyclical, you know, things that go on in the cell. And if we oscillate at a cycle of a calcium burst or something, calcium being the thing, there's a lot of information at like 15, 16 hertz around there. 60 is another one that you see a lot in the literature. But there is some new data coming out from some labs in cell culture and some theories that it might be a different type of resonance, right? Like, so maybe we're not just affecting electron spins. Maybe we're affecting, you know, nuclear spins or you know, some other explanation for why. But what we see is that you can much more easily change the effects. Like, so if a static magnetic field gives you an effect of like maybe 60% of them do this, right? That if you can get the right frequency, you can get, you know, a significant increase in the amount of what you're seeing. So I'm not sure the results would be different, but I think it would be easier to get them.
Okay.
Yeah.
Cool.
But that's a guess.
I guess this is some future work. That's
yes.
To invite you again in 2 years and see what the graphs
maybe 4. Maybe 4.
I'm confident 2 years it'll be fine.
Okay.
All right. Thanks.
Okay. So just a quick question from Chris. He says, maybe a silly question, but what are the exact differences between regeneration and healing?
So healing does require some new cellular growth, right? But generally healing is considered mostly doing what you need to sort of seal off the wound. And regeneration includes some component of replacing what's missed, right? So I think that's the easiest way to think about the difference. And it's not a silly question at all.
Thanks. I didn't know that definition myself, so that's good. And then finally, Pierre, I think the question you put in the chat is very similar to Hugo's question, but if you have a final question you'd like to close with, please go ahead.
I'm sorry. Apparently the postman's here.
We can't hear you, Pierre. Let's see, I think it seems like we can see Pierre waving, but we cannot hear him.
Maybe type the question into the chat.
So let's see. So Pierre's first question was also about the AC magnetic fields, but I think you answered. I think you just
AC is going to be much more interesting than this talk. Once we get into that data, I really think this talk will be boring and we'll never talk about it again. But
okay.
That's just what I think.
So yeah, Pierre says his question has already been answered. So with that, Wendy, and he also says thank you for a great talk.
Okay, thank you. I've really enjoyed this, guys. Thank you so much.
Yeah, thank you, Wendy. And this is the last talk of this term. So and at the end of every talk, and I've been doing this for the 5 terms throughout the pandemic, I've been promising the speakers a free meal and a lab tour next time you're in Cambridge. So it's building up to quite a number of people we're going to have to take out. But if you are around in the UK or Europe, please let us know, and we will definitely owe you.
Well, we have family over there, so they moved out of Cambridge, but so
okay,
I might take you up on that someday when we're allowed to travel.
Yeah,
we have magnetic fields that are a lot bigger than the ones. You know, you're talking micro Tesla. We have Tesla. So
yeah, I know. So
you saw your curve going up. If you want to put some of these things into some large magnetic fields, I'm sure that Pierre would be interested as well.
I don't know. I think you might explode them. It might be an interesting but very quick experiment.
You might grow some hydras.
Yes.
Oh yeah. Now, if we can turn planaria into hydra, you know, we've got a Nature paper. We're all famous instantly, forever. That would be awesome. Yeah.
Okay. Thank you,
Thank you, everyone.
Thank you, Wendy. Take care. This was super interesting. What a nice end of the seminar series for this term.